databases, an area that's really, really interesting. However, today he's going to speak about another very interesting topic, which is integrating diverse data sets, and especially in the context of his ongoing work with the Cancer Genome Atlas project, as part of his role as a senior research investigator scientist at Memorial Sloan Kettering. So thank you very much for joining us today.
So I'm going to talk about systems for managing, visualizing, and analyzing biomedical research data. And as Constantine said, my background is in computer science. I've had a couple of decades of designing, building, deploying production software systems in a variety of areas related to IBM, the company I founded, ChoiceMaker, and work we've done in computational biology. So a few years ago, I got bitten by the bioinformatics bug, and I've done work in the plant genomics at NYU and now cancer genomics at Sloan. So I'm going to present a very software-focused discussion with some case studies of systems we've built and data that we've analyzed. So I'm first going to talk about bioinformatics systems in general and talk about what would you like these systems to do for us at a very high level. And then I'm going to give an example of several pieces of production software that I've helped build. And then I'm going to talk about what people are calling platforms or workflow systems for bioinformatics and the promise that I think they may hold for the future. And I'll talk a little bit, if time allows, about my software engineering philosophy. So when I'm talking about systems here, I'm concerned about the underlying platforms that make it easy for us to do the rest of our work. So a lot of the time we focus on building analytical tools, whether they're for sequence analysis or copy number variation or mutation calling or other kinds of analysis. And that's very important, critical work. But what I'm focusing on today is once people have tools like that and data sets and loads of data sets and users, how can they use infrastructure to make it easier to service that entire community? So for at least purposes of discussion, I've highlighted three key goals of building these kinds of research systems. One is we want them to help us get easy access to reliable, vetted, best of breed analysis tools. Two, we want these systems to help us with data reduction and integration. So we really have very big, long data flow pipelines from high throughput data, clinical data, other things, microarray data, through to our final analyses and visualizations. And there's a great deal of data integration and summarization that happens in all of those stages. And a third key objective that I see is validation, which is something that we were just talking about. When you run your analysis through all of this pipeline, and you probably do a lot of backtracking and reanalyze and backtrack and reanalyze until you're done. how can you be sure that you've done the right thing? Or can you, at the very least, go back to the beginning and rerun the entire analysis and document all of the choices you made in terms of algorithms and input parameters and thresholds? And I think that that's really hard to do when we're writing programs on a day after day basis and discussing the results with other people and refining it. So it's really helpful to have systems that enable us to do that. So analysis tools, reduction and integration and validation. And finally, we need to maximize our productivity because there's way too much to do. I mean, people have loads of data now that's piling up for lack of somebody to analyze it. One of the things that I think is important for us to realize is that you know, we're already getting overwhelmed, and this is going to get worse very quickly. So this is an amazing curve, this red line from Lincolnstein's paper of last year, which is showing that with next generation sequencing, the exponential increase in productivity is twice as much sequencing per dollar every 6 months. So this is 3 times faster than Moore's law, which is the blue line here for disks, and is going to overwhelm us with data in ways that I think we can't appreciate. Plus, people are spending 22% more per year on this equipment, which is going to make it even worse. And so these systems are going to be widely deployed in many laboratories where people won't have the facilities to carefully and reliably process them. So also for purposes of discussion, we happen to have a pointer, by the way? No, that's a mess. I'll use my. For purposes of discussion, I'm breaking the code that we work with into 3 layers. Custom code, which obviously is the most flexible way to approach it. Platforms or workflow systems, by which I mean systems like GenePattern, Galaxy, Taverna, Kepler, VizTrails, and many others. And then portals, which is what people are starting to call web-based query and visualization systems. I mentioned a couple that I've worked on here, the MSK cBio Cancer Genomics Portal and a virtual plant system at NYU. But of course, there are many, many others. And I think they span this level of functionality in terms of the most flexibility at the bottom for somebody who's skilled at developing software to the easiest to use for people who aren't programmers at the top. And there are trade-offs between these things. So let me talk about 3 different systems that I've built, participated in building over the last decade, working backward in time. The cancer system at Sloan Kettering, the plant genomic system at NYU, and the machine learning-based approximate record matching system at ChoiceMaker. So the system that we've built at Sloan focuses on the Cancer Genome Atlas data, which is being collected presently. So the TCGA project is, for those of you who aren't familiar with it, is a very large, about a third of $1 billion in a decade and 15 research centers, detailed genomic analysis of cancer. We're looking at 20 different cancers in 20 different organs and about 500 patients per cancer for a total of 10,000 individuals. And across that across that set, we're getting copy number variation, mRNA and microRNA expression analysis, methylation, sequencing, and mutation column. So all that data gets pipelined to the NCI, then goes to the Broad, which normalizes it, and then we bring it into MSK, and we've put it into a system we call the Cancer Genome Portal that has many ways of querying and visualizing and analyzing this data, which I'll go into. So the first thing we need to do is handle this wide variety of inputs where there's a lot of the right word isn't inconsistency, but there's a lot of difference from one data set to another. So different data sets have different data types available, have different subtypes available, and so forth. And so we've created an interface that enables people to collect data while adapting to these changes. So they can look at the cancer study that they're interested in. They can pick which genetic data types they're interested in: mutations, copy number, expression, methylation. Protein level is available for a small number of genes in just, I think, one cancer type at this point. They can pick which cases they're interested in looking at, and we also create many subsets of cases according to the subtypes of cancer involved and the subtypes of data involved. So for example, for glioblastoma data, you can pick the people who have been treated or the people who haven't been treated. People are allowed to enter a set of genes that they're interested in, and our design point is for people who have one or two pathways of interest that they want to explore, and who will be entering a dozen or 24 genes that they want to look at. We also pre-create a set of genes, many sets of genes for known pathways, so that people can look at those carefully. So our key abstraction in terms of the way we visualize and present the data is genetic alteration. So we're interested in whether a specific gene has been altered, that is, its function is probably not working in a particular patient. And then we summarize that across many patients and across a set of genes. So this figure down here, for example, which we call an oncoprint, has a row per gene and compresses across its width all of the cases. In this image, we're showing 150 cases which have at least one gene altered, which is 73% of the 206 cases of data we have for glioblastoma. And the way the alteration is indicated here is in this small color coding. So the red and blue are copy number variations. The red indicates amplification of copy number, and blue is homozygous deletion, and the green indicates mutation. One thing you can see here, which we've seen in quite a lot of data, some other data sets even more noticeably than this one, some other pathways, is that there's a fairly significant amount of mutual exclusivity, by which we mean when the pathway is altered, only one gene is altered in that pathway. So you see a large number of patients for whom the only alteration in the TP53 pathway here is homozygous deletion of CDKN2A, a number of other patients that have these other three genes altered, and then a relatively small number of patients who have two alterations. The way in which we define these alterations is as follows. This is the default definition, and it can be altered, as I'll show you in a minute. But we say it's altered if the copy number is altered if there's been homozygous deletion or amplification. The expression level, which you don't see in this figure here, I'm not showing expression data, has been altered if the expression level is more than 2 standard deviations away from the mean. And what we do is normalize the expression level with respect to the distribution of expression values in the diploid samples. And that way one can talk about 2 standard deviations across the entire set in a uniform fashion. And the mutation, we're interested in functionally somatic mutations or germline mutation in susceptibility genes, so especially BRCA1 and BRCA2 in women's cancers. So what I'm going to show here is a 4-minute video screenshot of the system in use. This is an access-controlled system that contains the TCGA data. And one thing we've done to make things easier for ourselves is to offload username and password management to Google or Yahoo or somebody else. And there's a protocol called OpenID, which you can use, where you can say, go log into your account over there and tell that system that they can tell us that you do have an account over there, and then we'll get that protocol message and we'll let you log in here. So all we keep track of is, for example, people's Gmail address. So I'm going to click on this video is going to click on Google there, and it logs in without any login happening because this person is already logged into Google, and they've told Google that Google can tell us that we can access it. So this is the set of about 15 cancers that we have already. I'm going to show some colorectal data, which is currently being analyzed by the TCGA working group. And one of the things that we're finding interesting is with BRAF is that it's highly mutated, but it also has a huge variation in its number of mutations for the hypermethylated cases. So, I'm sorry, hypermutated cases. So there were about 29 of the 200, so about 15% of the 200 samples are hypermutated. And we see that 52% of the BRAF genes are mutated. 52% of the cases have BRAF mutations in the hypermutated cases. The vast majority of these are the well-known V600E amino acid change. And one thing that's an open question is whether the high mutation rate of BRAF is being causal with respect to the hypermutation or the other way around. Now we're going to look at the RB pathway and look at 4 genes in that pathway, IGF2, P10, PIK3CA, KRAS. This is the P10 pathway, I'm sorry. And we're going to adjust our definition for what alteration means. So we're saying the alteration here is just mutation, amplification, or expression more than 3 standard deviations above the mean for IGF2, PIK3CA, and KRAS, all of whom we expect to be overexpressed in if this network is dysregulated. And so what we see here is the oncoprint with these 4 genes, and the cases across horizontally. We see the light pink is overexpression, and the light blue is underexpression according to those 3 standard deviations. There are a number of different other ways that we can visualize data in this system. One of them is a set of plots that show expression as a function of copy number variation. So here we're looking at IGF2, and this is the expression z-score from 0 to 4, and this is the copy number variation, het loss, diploid, gain, and amplified. And what you see here that's striking is a bimodal distribution in the expression level of IGF2 for both diploid and gain samples. And so that indicates that there's something other than the copy number variation, which is contributing to these overexpression values. So that gives you I realize it's very quick, overview of the cBio portal. And there are 2 parts to this portal. There's this TCGA password protected version, and then there's a public version that has ovarian, prostate, glioblastoma, and 1 other cancer on it. So let me return to those 3 standards of objectives that I set for bioinformatics systems at the beginning. And let's try and evaluate these systems with respect to those standards, this cancer portal with respect to those standards. So the first, this is kind of a simple bar graph which is color-coded. And so the first standard is, does it have good, reliable analytical tools? And I would claim that, yes, it has some, but not a lot, because we focused on certain particular visualization and analytical tools. There were several others that I didn't show you. And it's somewhat tricky to add new ones. So I gave us, this is very subjective, I gave us a 1 out of 4 on that. Data reduction and integration, there I think we've done fairly well, because we've found ways of visualizing and combining in multiple ways the multiple dimensions of cancer data that we have, so I gave a 3. We really haven't done anything to help to address issues of validation, and our user feedback says that it's really helping their productivity a lot. So halfway to great. Let me switch gears now to the virtual plant genomic system, and I will tell you I'm happy to take questions or interruptions during the talk. So this is a system that we did in the plant genetics laboratory that Gloria Caruzzi runs at NYU. It's a system which has a nice feedback model, a little bit like some of the workflow platforms that I'm going to talk about in a minute, which is that it stores gene sets and gene network signaling pathways and makes it very easy to take one of those inputs and put it into some analysis tool and take the output of that and save it back for future analysis. So this is a system where we've seeded the data sets from a number of sources, from the gene ontology. You can take any node from the gene ontology and turn that into a gene set. From KEGG pathways, which you can do similar things with. From well-known gene families. We have data for Arabidopsis, rice, and it's being expanded to other plants. And one of the things we've done that I think is going to be very important in systems going forward, and that the cancer genomic system is working towards, is we have private accounts. So people can upload their own data, store it securely, share it with other people when they want, and that provides inputs to the gene sets and gene networks as well. So we have a bunch of analysis tools, and I'll show you a few of them here. Simple mathematical set operations is one. We have a very nifty tool that Tamara Tershikovic built, which is an interactive gene ontology browser. And you load a gene set into this, and it colors all the nodes in the gene ontology according to the level to which that gene set is overrepresented at that node, and then lets you navigate around it, pick the subset of your genes which are located in that node, and save them elsewhere. A third tool which is really neat, which is built by Chris Pultney and Dennis Shasha and others, is SunGear, which is basically a Venn diagram tool on steroids. This lets you take an arbitrary number of sets of genes and look at all distinct intersecting subsets. So, for example, to make it simple here, I've shown an illustration where there are 3 input sets of genes represented by the 3 vertices of the triangle, and each of these white circles represents a set of genes that are present exclusively in some subset of these. So this circle represents the genes that are in here and here. This circle in the middle represents the genes, represents only the genes that are in all 3 of the sets on the vertices. And this can expand to a polygon of arbitrary numbers of vertices. It also has a lot of other functionality for statistics and so forth that I'm not going to show you now. So the third system that I want to talk to you about is our ChoiceMaker Approximate database record matching system. This, just the history of the company, I co-founded it about 11 years ago. The company made it for about until the recession started, and we had NSF financing and 15 production places using it, including the cancer repository in Sydney, Australia, and the immunization database maintained by New York City. The problem we're solving here is one of database records that lack unique identifiers. In the immunization registry, they're not allowed to collect social security numbers or another unique identifier because they don't want to discourage people who don't have social security numbers from getting immunized. So they collect a whole set of demographic data about each child. Name, address, parents' name, date of birth, medical record number maybe. But unfortunately, for a lot of children, don't see the same pediatrician each time they get immunized, especially lower income. And therefore, they don't get the same demographic data collected. And so, before we started working on this database, there were information about 1 million unique children in the system, but they thought they had information about 2 million children because there were so many duplicates. So we built a general purpose approximate database record matching system that has 2 phases. The first phase is called blocking. It's just a term of art, but basically it means getting many possible matches out of the database to the input query, so that you don't create any false negatives, but you winnow down the size of records you need to look at by several orders of magnitude. And then a machine learning based matching system, which we use maximum entropy because it has very high explainability, and then produce outputs that has a three-part output. It produces a probability that an input query record matches one of several records that are similar in the database, and for each pair says this is a probability of match, and then sets some threshold so that if the probability of match is below a certain threshold, it definitely differs. The probability of match is above a certain threshold, then it definitely matches. And in the middle, it's something that we recommend for human review. So in our production systems, we were able to get false negative and false positive rates of under 0.3%. This was very good and was just as good as was obtained manually by clerks working, for example, for New York City, when they would have two clerks independently reviewing pairs of records and trying to make decisions about whether they matched or differ. Their error rate was that high or higher. So we trained the system to do as well as people could do. One of the things that we had, which was very neat, is a graphical user interface for ML model development. And basically, we built a custom language which enabled people to describe matching features, which we called clues. And we had a multi-tabbed integrated testing and training environment that would let you take a training set, see how it fires on the clues, decide which clues make sense to keep, refine the training, compare it with a test set. And in the test situation, we produced data like this, where the probability goes from 0 to 1 of match over here. The green bars indicate records that were marked match by the clerks, and the red bars are records that were marked differ by the clerks. And this is a vertical log scale of the number of records. So the vast majority of records that were marked differ or marked match were scored as very low probabilities or very high probabilities. And the ones in basically this range here, which is about a dozen records, were the ones that we, in this particular model and data set, marked as needing human review. So that summarizes these three systems that we've worked on over the last decade. And now, as I mentioned, I'd like to shift gears and start talking about bioinformatic platforms. So the architecture and premise of bioinformatic platforms like GenePattern, Galaxy, Taverna, and so forth, is that you can take a system, put a bunch of bioinformatic tools in it, maybe hundreds now in these systems in different classifications, and make it easy for non-programmers to make their data accessible to this system, access their data through these tools, run the tools, produce the output, and then notably save the output from every execution so that that can be used as data for the next execution, and so that the history of the execution can be reviewed. My overall assessment of these systems are they're a very good idea that still needs a lot of work. And the reason they're a good idea is because they create a common platform for achieving a lot of the objectives I set. And the reason they still need a lot of work is because these are very complicated systems. When you think about it, they're distributed operating systems for bioinformatics. And there's a great deal of functionality at all levels in terms of data access, access to computation, especially parallel computation, access to tools for processing histories, and so forth, where there's a lot more work to be done. And that makes a lot of sense when you look at other kinds of systems that aren't in bioinformatics and finance and so forth, that have had hundreds of person-years of effort involved in their development, whereas these have had dozens. But let me give you a quick overview to Galaxy if you've never seen this. This is work by groups at Emory and Penn State. The concept here is that there are a bunch of tools that you can choose from, and once you pick a tool, it gives you an input form to fill out, and you select those input parameters, and then once you execute the tool on that input, it produces output that you can look at and creates a history item in the stack of executions that you're developing. And then you have a history that you can work with so that you can select any one of the items in the history and look at the input and output for it, change its parameters, and run it again. So the really powerful concept here is that you have these histories and these workflows as first-class objects that you can process. There are a lot of good things that you can do with histories: save, share, publish, import, export them. You can convert them to a workflow, which would be a picture like this. This is a data flow is going from left to right. You just can't see the arrows in the screenshot. This was a simple demonstration of putting together 6 different modules to find, that will let you find the 5 exons with the most SNPs on chromosome 22 using the UCSC data. Once you have a workflow like this, you can consider it to be a single program, and you can run it on data sets. You can import, export it, share it with people, edit it. So I've spent some time recently looking at 3 of the major platforms: GenePattern from the Broad, Galaxy that we just looked at, and Taverna that comes from University of Manchester in England, and trying to compare them on these criteria that I described: access to tools, data reduction and integration, validation of results, and maximizing productivity. There's a little too much for us to look at all of these right now, but let me pick about 4 of them to highlight and talk to you about those. With respect to extensive libraries of analysis, everybody's doing quite well. Taverna has an interesting model, which is it has a functionality that lets you search the internet and look for additional tools that are exported as web services that you can send data to. So I think that's an interesting model. Whoops. With respect to NGS tools, GenePattern is somewhat along the way for that. A lot of that's in production, and Galaxy's done a lot of work on that in the last year. With respect to access to public data, the Taverna searching lets you do that as well, and Galaxy has a lot of tools for accessing, for example, all the reference species databases, but there's still a fair bit more to do on that. There's not a lot that these people do in terms of differential analysis, and I'll talk about that a little bit in the next couple of slides about things that I think would be interesting and exciting to do. One of the things that I highlight as a problem for Taverna is that they're not a accessible through the internet. So they've designed a Java program which makes it easy for them to produce a good GUI for building workflows. But that means you have to download the Java program and install it to run their system, whereas both GenePattern and Galaxy are web applications, and if you want to run it yourself, you install your own web server. You can install it locally, install it on your cluster, install it in the cloud. And I think that, given software trends these days, there's loads of functionality to be able to write programs in the browser, or JavaScript, ActiveScript free. It doesn't make sense to force somebody to download a Java program just to use the system. There's been a fair bit done on parallel and pipeline execution and cloud execution by these systems, but there's still a good deal more to go. One of the things I did, and this is just fun, this isn't publishable, but was to try and get a sense of how heavily used these systems are. And that's simply because in the software world, you want to work with systems that are heavily used, that a lot of people are contributing to, that are growing. That's the network effects that we deal with. And if you're picking something that's a winner, then you're going to ride that wave. If you're not, then you'll find something that doesn't provide you with the functionality you want, and eventually you're going to have to switch. So I did a bunch of Google searches, and Galaxy and Taverna and Kepler have pretty high scores. This is the social network analysis with Google+, LinkedIn, and Twitter. These are there's a nice little web API somebody put so you can issue queries to them, and they'll tell you how many references there's been to a particular URL across all of these websites. And there you see it pretty much echoes the same data that the Google data had. There's a lot of scores for the Galaxy work, which these are 3 separate sites they have, and a lot of scores for these 3 separate Taverna sites, and less so for these other sites. I tried to do this on the citations in Google Scholar, but it won't let a machine access it. So I want to talk about a couple of interesting and exciting ideas I have for further analysis of workflows that come out of these patterns, out of these platforms. So suppose you've run a big workflow, and then the input changes a little bit. For example, you might want to just adjust one of the threshold parameters and see what happens. You'd like to be able to rerun the workflow with minimal additional computation, especially if it's time-consuming. So imagine you illustrate the workflow this way. These are the inputs. P1 to P4 are the modules that are part of the computation, and O1 is the output. If we have a history, so we have one that we've already run, then we can look at it like this. Now suppose you want to change this input parameter. Well, then you know that what passes on that edge is changing, but you don't know whether what passes on the edge between P1 and P2 is changing. So you can list that and all these other downstream edges from it as unknown. Then you can execute P1, and in this case, I'm saying that the output for P1 changed, in which case you're now going to need to execute P2. But if the output from P2 doesn't change, then you don't need to execute P3 or P4, and you in fact know that the output from the whole system won't change by the change of this input parameter. The simple algorithm is you run any program whose inputs have changed and continue that until you've decided whether all the inputs have changed or not changed. So this is an interesting underlying functionality that we could use to study the stability of bioinformatic analyses. There's a lot of work that people are publishing in bioinformatics depends upon somewhat arbitrary thresholds, whether it's the number of bases they're cutting off the ends of reads because they want to get rid of the noise, or some threshold in blast hits. And we really would worry about analyses if their final analysis result depends a great deal on these thresholds. We'd like to know that they can change the threshold a lot without changing the output very much. So for example, there are some analyses that we've seen in cancer genomics where people thought they found a lot of new oncogenes, a lot of new genes with significant mutations, whereas it turned out slight changes in the assumption of the background mutation rate would render many of those mutations not statistically significant. So in general, what we want to do is look at the partial differential of the output with respect to each of the input parameters. So we'd have a picture like this, some big input data sets, a bunch of data parameters, and a bunch of processes. And we can use the tool in the previous step to take a bioinformatic analysis and do a thorough stability study of it, which I think would be very valuable. I mean, I'd love to see a world in the future where when people are publishing things, they publish the software in some usable state so that it's easy to make these kinds of analyses. Finally, I thought I'd talk a little bit about my software engineering philosophy. This is very much largely kind of motherhood and apple pie for agile software development. I believe it's important to release software frequently and get feedback from real users. Have iterations with, that are constrained with deadlines. Do a lot of quality control. Make sure that developers have excellent tools, software tools, computers, displays. I believe in groups where everybody writes software, and it's critical to have a vision about where you're trying to get and keep an eye on what the competition is doing. And I thought I would conclude with a bunch of writing which I found inspirational in terms of software quality, both for small-scale concepts about how to write good programs to large-scale concepts about how to structure software teams so that they can work successfully and collaboratively. There's a lot of people who've contributed to the work I presented today. We have a team of about 8 people at Sloan who are working on the Cancer Genome Portal at various levels of commitment. Nikki Schultz is our kind of lead biologist, and Chris runs the group. The work at NYU has been done by a team, and likewise for all the other systems I presented. I'd be happy to take any questions. Yes. To what extent does it, what can you mirror? Right. That's a very good question, and that's an issue with we haven't felt that our portal has really supported well enough. When you look at the two-dimensional grids of genes versus cases, and you just see a gene that's labeled as mutated or not mutated, for example, can you verify that? And one of the things that we're doing, because people haven't been able to drill down and verify that, is make it possible that somebody can click on that gene and open IGV on the data so that they can see why a mutation was called there or why there wasn't a mutation called there. In fact, people have realized just quite recently with the data that there's been a bunch of undercalling of mutations, especially because solids have been showing deletions at the ends of some of the reads. And the way the calling has worked has been to compare the normal with the tumors, and we're seeing a lot of false deletions in the normal data. It led the calling program to not call mutations at those points. So I think that our portal, I mean, we recognize that our portal hasn't provided enough transparency for the whole data flow analysis. I think that the workflow tool platform have the promise of providing better ways of doing that, because you can work backwards through the history and look at the various steps and see how the data looked at those steps. Although they don't have the support yet to do that easily. So for example, I can't do, if I put my tool, which actually I would like to do, put that visualization stuff into GenePattern or Galaxy, and then they take care of getting the cancer data in there so people can look at it. There isn't easy, for example, drill down to say if I look at a particular data conclusion and I look at the particular pieces of data that led to that at each step along the way. There's some very impressive software in the financial world called Ab Initio. You never would have heard of it as a very secretive company. They sell their software for $1 million, $2 million a shot to banks. And they have this amazing data provenance graph modeling stuff. So any piece of data you want to, you can look at and you can see what the previous data sources were. Systems like that, I mean, they don't have genomic data, so they don't have hard data set size. But they actually, the metadata to keep track of all of the data dependencies ends up being about 90% of the data in the system for exactly those kinds of things. Well, they do have version control systems. So for example, if you're writing software for GenePattern, and every time you change your module and install it again, you get a new version number. But there's a difference between having a version control system and being able to drill back through the entire dependency analysis on a certain data element. That's another level of functionality. That's right, yeah, and it's unfortunate because the work that Chris Pulteney did for the, what's it called, Venn diagram on steroids is a wonderful tool, but nobody has the time to integrate it somewhere else, at least not today. So I tell him I'd love to have him do it, and maybe someday I'll find a student who I can. And Chris would be happy to have me do it. I don't find either. But absolutely. We could certainly load it with the human genome, and but you know, it's definitely yes. You just have to change the data, but there end up being a lot of consequences to that. You have new gene families. You have new some other things change as well. So plus, you need to get a user community for it to justify the effort in doing that.
You mentioned
Yeah, the virtual plan. I'm not sure what you mean about complete, but the ways in which the system can be changed are, there are several ways the system can be changed. One is you can create your own installation and then add your own programs to it. Really straightforward. Or if you have a tool that you'd like to be part of the public site, you can submit your tool there and ask them to install it. Or you can share tools from other people. There's now a tools repository that you say where you can add things. So, I mean, complete is a lot, but I think there's underlying functionality to add new tools to it in a fairly straightforward way.